Oh, good morning, students. How are you all? So, here today I am going to explain the continuous already I taught for monohybrid cross and so many things, law of segregation, law of that is for inheritance, law of unit characters, law of dominance, everything I taught of you all. So today I am going to explain the part 2 of that class 10 heredity and evolution. This is part 2 I am going to explain. Dihybrid cross. Di means two hybrid cross. We are taking tall with round seeds. Tall with round seeds. You know that? Okay. T, capital T and R are round seeds. This is seeds. We are taking R and R. Don't get confusion. So okay. After that, TR again TR. We are taking two groups. Here we are taking the dark with the dark means you know that is for short tree. That top with a wrinkle, we know that very wrinkle means how to write. Wrinkled. So here the small t, that is small t, and small wrinkle, that is a wrinkle r. That small t to 1r. Here we are taken t to t. We are taken that is for. After that we are comparing this part and this part. Anyone can take this part or this part, both are same only. First capital T we are taken, that is small t we are taken here. See, capital T, that is small t, and capital R and small r. That is the F2 generation we are taking here. So what we are taken here? That is what it is a we get if we gametes where that is combines all are tall with round seed, and that is for F1 generation. That is for F1 generation. Next, I am going to explain that is F2 generation. In the table I will explain here. Now I am going to explain the F2 generation here. That is F2 generation here. So here we can take the table, this row, and only this first row and first column. How multiply this into this. T, see here, this into this. 1T, 2T, 1R. Again similarly this into this. 1T, 2T, 1R, capital R, small r. Similarly like that. 1T, small t, 1 capital R, right? Again this T and big T and small t, capital T and small t, capital R and small r. After that, similarly, second to first, this into this here, taken this into this here, this into this here, this into this here. After that, third one, this into this here, multiplied, this into this here, this into this here, and this into this here. After that, fourth one, this into this here, this into this here, this into this here, and this into this here. See, first, now we are taking the phenotype, capital TR. How many capital TR we are taken? We have to see this part. First, capital T and capital R, 1. Capital T and capital R, 2. Capital T and capital R, 3. Capital T and capital R, 4. Capital T and capital R, 5. Here no, capital T and capital R is not there. Capital T and capital R, 6. Here no, capital T and not capital R. Here capital T and R, 7. Capital T and 8. Here no capital T and capital R. Here one is 9. Total how many we got? Here is not there. One capital T is there, but small r is there. Here small r is there, capital R is there, small t is there. That is 9. Understood? Now capital T and small r we can see. Capital T and small r. 1. See, 1. Capital T and small r not there. Capital T and small r you see. Capital T and small r is there. 2. I will write on the top. 2. Capital T and small r 3. Capital T and small r 4. Capital T and small r 5. Capital T and small r 6. Capital T and capital T and small r 7. Capital T and small r no. Capital T and small r how much over? 7 after 8. Capital T and small r 9. 9. Capital T and smaller, not there. Capital T and smaller, not there. Total ratio, how much we are getting? That is, we are taking only for exactly capital and small, only 1 hour, small 1 hour. Capital T and small, only 1 hour, you see. We have to take, there is 1, capital T and small r, that is 2, and capital N 3. Here 2 is there. No, we should not take. This is also, we will get the ratio is, that is like that we have to take, that is for 3 is to. Next, small t and capital R, we will get the ratio from if you find out 3. And small t and small r is 1 only. This we are taking the ratio of that. Understood? So, we have taken the capital T and capital small r. See here, capital T, where is capital T? Capital T and small r. You see one here, right? And capital T and small r. Where is that? Capital T and small r. This is 1, 2, 3. We got this 3 ratio and 3. That is for F2 generation we are done. Next, I am going to explain the sex determinant. Now I am going to explain the evaluation. It is a gradual sequence of changes that took place over years and lead to development of entirely new species. So we know that evaluation, we must be wondering that we should have some evidence to prove that occurrence of evolution. Evolution means from the olden years we are taken from the Charles Darwin. Charles Darwin has prepared, he has the origin of species he has done. So from monkeys to small babies of monkeys, and monkeys to that body of that, it is bending again to the both hands, both have 4 legs, 2 hands and 2 legs as towards on the floor. Here they are taking onto the land. And after that, little bit increasing to the stand of the bending, again in bending and standing like that. It was standing as a man. So Charles Darwin has prepared the origin of species. In that he has studied about detail in that all those things. So here we are getting the evolution of that. That the different evidence of we are getting the fossils. Fossils means we know that. Fossils means dead animals or dead bodies. We are collecting from the dead. So the dead remains of the plants and animals that got buried under the earth. It was buried into the earth million years ago. That study is called fossils. Understood? We are getting the lot of years just as a mummy body. You know that we are getting from the buried in the soil, and we are taking now the bodies. We are measuring the lifespan, how many years that they before they died and how many years he was alive on the earth. That and all we are finding, that is for fossils. Understood? Fossils reveal the order of origin, that is for protozoans or fish, amphibians, reptiles, these birds. What? Fossils reveal the order of origin, how the protozoans are given: fish, amphibians, reptiles, birds, mammals. Understood? Archaeopteryx. Next I am going to explain that Archaeopteryx. 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 What do you mean by Archaeopteryx? It is that resemble birds as well as reptiles. It is a resemble birds as well as reptiles. We can say that is for link between reptiles and birds. How? Assembling means breeding of the animals with the birds. That they are taken that Archaeopteryx. That is the word they are using for that. They are using to cross breeding of the animals to birds. That is nothing but Archaeopteryx. Understood? Homologous organs. What do you mean by homologous organs? are those which have same structure but different perform functions. Structure is same. Just you see, one small parrot and macaw, they both have same structure but they have different functions. Macaw will fly more. It will fly more. It will more places it will go. It will high. But parrot will not fly maximum of that. Right? Eagle and another birds we have to take. But face are same but different function. If you take cat and cheetah, but they both are species are looking same only, but they have different function. Cat is little bit different. They have different functions. Understood? So like that is there. Four limbs of man and cow. 4 limbs of man and cow also. That is difference. Analogous organs. Analogous organs are those organs which have different structure but perform same function. They have different structure but they will perform same function. Example, wings of birds. Wings of birds, you see, all the birds are different structure is there, but the wings are flying only, right? And insects also, and different wings are there but different functions to fly. Different birds have different functions also, it will fly, but they will fly more on the top. Some birds are going not top, only the 45 angle it will fly. Understood? And vesical organs are those which function in our ancestors, but now that not functions. Example, membrane, pinna muscles and wormy functions or appendix of man. But different necessitating function means, so necessitating membrane in human it's not function, and it's blind its function. It is not functioning but it is a blindly. Understood it? Birds in birds its function. In human not functional, which necessitating function membrane. In human not function. It has not function, but in birds it function. Understood? Pinna muscles. It is a present in tail of bone. Where it is present? Pinna muscles. It is present in tail of bone. And vermiform, it is also present in tail bone. Understood? It is present in tail bone. Embryology. Embryology. What do you mean by embryology? It is a study of development of embryo. This study reveals that all organs of different species have common ancestors. That is for chimpanzee, hen, lizards. In first stage, they all have look like same. How they will look like? Same. Ancient means they look like as a same. But chimpanzee, hen, lizard, when they will born in the stomach, inside the stomach in the mothers, they look like as a same. Just look like as a same, like that. Like that it will look. Right? In the cell. Speciation. It is the formation of new species pre-existing. Pre-existing ones means pre-old ones. Understood? That is for formation of new species will get formed. That is for, it is one of the pre-existing one. It is pre-old ones only. Allopatric speciation. Allotropic means a new species may be formed from the population occurring in different areas of distribution. Understood? Allotropic means it is the new form of a species may be formed by the population occurring. It increased the number of areas. Area by area it was increasing that is for distribution. Sympatric speciation. Sympatric, a new species formed from the fragment of population in the same area due to the mutation. fragment of population in the same area. Micro-evaluation. Micro-evaluation, micro means small evaluation. It is the formation of species due to small but significant changes like genes, mutation and recombination. Genes means you know already, it is changing of the characters of the from mothers and fathers, and some other characters will be formed into the genes. That is for mutation of recombination we are done into the species of the new ones. Understood? Now macro evaluation. Macro means it is the larger, it is the development of different genera and taxa. That is for macro means it is a large one, we are producing the organs in that. So many characters are getting from that. So that is why we are getting into the evaluation. Next, allele. Allele means what? Allele means the two contrasting genes corresponding to the same character. Just as you take one TT, that is for same contracting. Height we can measure as a example height. Measuring from father to, that is for baby, that is for we are taking the height only. That is for nothing but, see once again I will repeat. Two contracting genes corresponding to the same character. Two correspond, that is for taking for the corresponding from the mother and father, and it forms as a father only. It genes has been converting into the character, that is for height we will observe also. Understood? Homogenetic, it produce some type of gametes. What it will produce? Which produce same type of gametes it will produce. If the given just you have to take TT, it will produce same gametes. That is we are taking for that same TT. Understood? And heterogametes, which produce different types of gametes. It produce different types of gametes. So this is the topic. I have done all those things explanation. If anything required, you can find out in the textbook also, and you find out that there are some other textbooks, or you can refer the other books also. You can get all the knowledge. Okay. Now this is your topic. I have done this topic. This is the part 2. I have finished on that. That is for heredity and evolution. Okay.